This is the Professor, CBMD PhD, and today's Monday, March 24th, 2025. It is 6:20 and 38 seconds, p.m. Eastern Standard Time, and today's research, eukaryotes. So let's jump to it here. The fact that eukaryotes arose from teams of prokaryotes is unquestioned in biology. Exactly how this happened, however, is still a subject of much debate. Before we explore what is not known, let's establish the facts that all scientists agree on. Eukaryotic cells are so named because one of their most distinctive features when viewed under a microscope is the nucleus. The name eukaryote is derived from the Greek eua, meaning true, and keri, meaning kernel. Prokaryotes are therefore said to have arisen pro or before the nucleus arose. It is tempting to think that the appearance of the nucleus marked the onset of eukaryotic life. Yet, the first organelle to develop was the mitochondrion. This occurred approximately 1.8 billion years ago, almost 2 billion years after life appeared on Earth. Eukaryotes have on average 4 times more genetic material than bacteria. The largest known amount of DNA in a eukaryotic cell is 10,000 times more than the largest known in a prokaryotic cell. This means that eukaryotic cells contain more genetic information than prokaryotes. Eukaryotes have over 1,000 families of genes that are absent in prokaryotes. If we use the cell as a busy city analogy, we can say that the library in a eukaryotic cell has both a larger and more complex collection than the corresponding prokaryotic library. This is an important consideration because it helps explain why eukaryotes are so much more structurally complex than prokaryotes. They have more information to work with. Eukaryotic cells are on average 10 to 100 times larger than prokaryotic cells. While size alone does not necessarily confer any special advantage on eukaryotes, their larger size does reflect their increased structural stability, which can be a tremendous help. Borrowing from the busy city analogy, we can say that skyscrapers are far more complex than mere stacks of small buildings. They require specialized structures that allow them to remain functional under conditions that would break simple clusters of small buildings or cells apart. Much remains to be explained for us to understand eukaryotic evolution fully, but common themes are emerging. For example, based on careful examination of the sequences of DNA, the nucleus, and the small amount of DNA remaining in the mitochondrion, biologists have determined that the source of most nuclear DNA is an archaeal ancestor, and mitochondrial DNA traces back to a form of bacterium called an alpha protobacterium. This strongly suggests that eukaryotes arose when an archaean host cell internalized a bacterium that was capable of generating more ATP than it needed to survive. This bacterium became the mitochondrion. This explanation is known as the endosymbiote theory, or endosymbiosis. In 2015, scientists discovered an archaeal organism called eukaryotes, which is strikingly similar in genetic makeup to this proposed archaeal ancestor. All eukaryotes share this genetic ancestry, strongly suggesting that the formation of the mitochondrion only happened once during evolution. Exactly when and how this happened are currently topics of debate. We know this occurred before the nucleus formed because the structure and molecular composition of the membrane surrounding the nucleus, called the nuclear envelope, and all eukaryotes are more similar to bacterial membranes than to archaeal membranes, and the source of this bacterial membrane is most likely the ancestral mitochondrion. An alternative hypothesis is that the early eukaryote internalized a second bacterium that contributed to the nuclear membrane. We also know this event triggered the division of labor that gives rise to our first principle. Once internalized, the mitochondria no longer had to move through its environment, e.g. swim, find food, or defend itself against attack. This allowed it to specialize in creating ATP, and turned the host cell no longer had to commit its plasma membrane to ATP synthesis. Instead, it could further specialize to form stable attachments with its surroundings, including neighboring cells. Teamwork between archaea and bacteria was thus immortalized in the form of a new biological domain, eukarya. A second common theme in the theories of the origin of eukaryotes is that mitochondria appeared well before they started, functioning the way most modern mitochondria do. Modern mitochondria rely on oxygen, O2 gas as the final electron acceptor when they convert food energy to ATP. Depriving aerobic eukaryotes of oxygen can be fatal. But at the time the first mitochondria appeared, atmospheric oxygen levels were much, much lower than they are today. What, then, were mitochondria using instead of oxygen at that time? Again, the power of genetics helps us solve this riddle. Bacteria that live in oxygen-poor environments today use proteins derived from genes that are very similar to those mitochondria used to create ATP. But these oxygen-poor bacteria substitute oxygen with other biological molecules, or even simple hydrogen ions. Researchers argue that if modern bacteria are using the same genes as the earliest mitochondria to make ATP, in the absence of oxygen, the first mitochondria could have done so as well. This means that the earliest mitochondria were adaptable enough to use electron acceptors other than oxygen, and that they later acquired the ability to use oxygen when it became available. Some modern organisms can use both oxygen and its alternatives. Because we have no direct proof that this occurred, this point remains open to some speculation. A third element common to all models of eukaryotic evolution is that the first eukaryotic cell had roughly twice as many genes as its prokaryotic neighbors, and because many of the archaeal genes initially performed nearly the same functions as the corresponding genes in the bacterial ancestor, many of the genes in the eukaryotic DNA, the collection of all genetic information in an organism, is called its genome, or redundant. In evolutionary terms, this fact is important because it provides a tremendous opportunity for the eukaryotic cell to mutate its genome without fear of dying out. Put simply, the first eukaryote could tolerate mutation of one of its genes as long as its cousin gene remained intact and functional. This genetic redundancy meant it could afford a lot of mistakes. This capability allowed for a lot of variety, including new genes not seen before in prokaryotes. For example, over 280 families of new genes arose during this period, and they contain both archaeal and bacterial sequences, demonstrating that they are the product of fusion between multiple genes. Not all genes survived the selection process. Modern eukaryotes are missing some of the genes in their ancestral cells, and some bacterial genes have moved from the mitochondrial DNA to the nuclear chromosomes over the past billion years. Most modern eukaryotic genomes even include some of the failed non-functional genes inherited from these turbulent times. Exactly how this proliferation of genetic diversity unfolded over time remains unclear and is open for interpretation based on different models of eukaryotic evolution. Fourth team, in response to natural selection, prompted yet more division of labor over time, aided by careful analysis of the genetic history of the proteins that are associated with specific organelles and cellular functions. We can now propose a reasonable explanation for how organelles and other specializations developed. For example, the organelles and vessels that comprise the endomembrane system, covered in detail, arose from the mitochondrial membrane, while the molecular machinery responsible for controlling membrane trafficking within the system arose from the archaeal ancestor. Extension of the nuclear membrane gave rise to the endoplasmic reticulum. When cyanobacteria acquired the ability to harness sunlight as an energy source, they released molecular oxygen to the atmosphere as a product of photosynthesis. Some eukaryotes later internalized these cyanobacteria, giving rise to chloroplasts, and eventually plants. When the levels of atmospheric oxygen rose high enough to pose a danger from reactive oxygen species, e.g. superoxide O2 and hydrogen peroxide H2O2, which can damage DNA and proteins, the mitochondrial membranes budded to create the peroxisome, an organelle that neutralizes these dangerous compounds. The threat of damage from oxygen radicals may have also promoted the development of other organelles, such as the nucleus, which protects DNA from damage. Because these intermediate stages no longer survive, we cannot definitively determine the exact sequence of events that gave rise to these eukaryotic specializations. The fifth theme in eukaryotic evolution is that, regardless of how many times the above events occurred, only a single eukaryotic precursor survived with all the elements shared by every subsequent eukaryote. This organism is now appropriately called the last eukaryotic common ancestor, or LECA. Remember that LUCA gave rise to LECA while its historical existence is unquestioned. It is unlikely we will ever find a living example of it. As we learn more about the evolution of eukaryotes, Our definition of LECA will likely evolve as well. This video was created for educational purposes only. Codename Will, Professor Christopher Brunette, Leader of Research, Scientist, Ufologist, Psychic, Computer Programmer, Computer Technician, Philanthropist, Writer, Mathematician, Artist, Photographer, Film Producer, Chef, Animal, and Nature Lover. Passion, World Peace. Hope you enjoyed listening. Until next time, have an excellent day. This is a Professor CBMD.